So now on with the show. I want to introduce Dr. Carson Holt, one of the makers of Maker, as it were. And Carson, I'll let you go ahead and take it from here.
All right. So my name's Carson Holt from the University of Utah. I'd just like to say thanks to Chris, Anna, and Glenn for inviting me to give this i5k webinar. I'll be talking to you about automated genome annotation and analysis. So to begin, I'd like to start off with one of the most heavily used slides in bioinformatics presentations. This is the NHGRI graph of how genome sequencing costs have dropped over the last almost 2 decades. The graph's in log scale, so this linear decrease at the start of the graph is actually a logarithmic exponential drop in the cost of genome sequencing. And in around 2007, 2008, costs just plummeted in an exponential form. And today, you can sequence what would have cost tens of millions or hundreds of millions of dollars to sequence for just $1,000 or a few $1,000s, depending on how much sequencing coverage you want for your assembly. And as can be expected, as prices fell, the number of genome sequencing projects underway exploded, as you can see here. The overlaid graph is from the Gold Genome database. The database tries to track info on all genome projects around the world. You'll see separate lines for each domain of life: bacteria, metagenomes, eukaryotic, archaea, virus, etc. And one of the things you'll notice is that since about 2015, the number of genome projects underway in all domains has essentially doubled. As many projects have gotten underway in the last 3 years as the previous 15 years combined. So with just eukaryotic genomes, as you see here, over 20,000 projects were started in just the past few years. If you assume 1,000 genes per genome, that's over 200 million new gene models that need to be annotated, stored in databases, and distributed. Then, of course, there's quality control and updates and maintenance to the imperfect models from the hundreds of millions of genomes already in databases from the many years previous. And now let's go ahead and skip on to a just genome project overview of a single individual genome project to better understand the importance of genome annotation. Let's say I, as a researcher in a small laboratory working on an emerging model organism genome, some species of cone snail, for example. And because research into this organism could reveal any number of important scientific insights, and because sequencing now is just ridiculously cheap, and as you've seen from the previous slide, sequencing is no longer the true bottleneck to genomics research. It's more the downstream analysis and annotation. I am going to, after sequencing, annotate the genome. So what is genome annotation? Annotation is basically the process of defining features of the genome. For example, annotations can be structural, identifying things such as introns, exons, start codons, stop codons, UTR, or the CDS regions of a gene. Or annotations can be functional, identifying things such as gene function, a location of expression, or protein domain content, for example. Now, once I've annotated the genome, I want to put that information into a database, something like Flybase or Wormbase, and from there, annotations can be distributed to guide downstream experiments so that I, as a researcher, can then query the database to extract things like a protein sequence that I can use to design experiments. I can do PCR-type experiments. I can perform expression-type analyses, protein domain analyses, or do more complex experiments like gene knock-in, knockout-type experiments using the annotations from that database. And if the annotations are correct, then my experiment should succeed and my career will advance. But if those annotations are incorrect, then my experiments will fail and my career will go in a different direction. And it's important to know it doesn't matter how hard I work, because incorrect annotations poison every experiment that uses them. It's a lot like the popular saying, all models are wrong, but some are just more wrong than others. So quality control is paramount, and having an evidence trail attached to each gene annotation is critical for the downstream researcher to know just how much confidence to have in each part of the gene model. So to help with this issue facing many research groups, our lab developed Maker. It's an annotation pipeline and genome database management tool for second-generation genome projects. I'm going to spend much of the rest of the webinar describing Maker and using Maker to describe the process of genome annotation, primarily because that's what I know and it's the algorithm I helped develop. So Maker is easy to use by design. It's designed to be able to be run by an individual with little bioinformatics experience, likely a graduate student in a lab. It can be run on a laptop or desktop computer. It can also run on high-performance computer clusters. And while it can run on any machine, we do recommend running it on larger servers just for runtime reasons that I'll explain later. Output of Maker is compatible with other popular annotation tools out there. For example, you can use Apollo to manually manipulate Maker annotations. You can use programs like JBrowse or gbrowse to view those annotations online, or you can use other GeneMod tools to perform other types of downstream analysis with the gene models and annotations. And of course, Maker is a free and open source tool for the academic community. So now let's take a look at what Maker actually does. The steps Maker uses are going to be similar to steps you might find in other annotation pipelines like Grameen or Nomon. So understanding annotation steps used by Maker will help you better understand what assumptions many tools put into building the gene models you see and work with every day. So here I'm going to use this view from the legacy Apollo desktop version of Apollo. It's an annotation curation tool. Here we have the genome sequence running left to right with the coordinates on the genome assembly marked here below. In this dark panel is where evidence alignments against the genome are going to appear. And in this light blue panel, the gene models or gene annotations will appear against the genome. And these green and red dots are just start and stop codons in the different reading frames throughout the assembly, so you can just ignore those for the most part. So the first step to Maker is to repeat mask the genome, to remove things like transposons, shown here in red. These can end up contaminating your final gene models because many classes of repeats encode real proteins. And those open reading frames can confuse gene prediction algorithms, so it's convenient to just mask those regions out so gene predictors cannot even see them. Masking also helps avoid spurious alignments of transcript and protein evidence that tend to align in regions of simple repeats like strings of A's or strings of T's and create false regions of homology. So the next step in Maker is we run ab initio gene prediction algorithms. These are programs like GenMark, SNAP, and Augustus. These algorithms use mathematical models, often HMMs, hidden Markov models, to look for expected patterns of gene structure. In the simplest form, these might be probabilities representing the transition from an intron state to an exon state, depending on the context of the sequence in a given region. And Maker runs these algorithms for you, and because they look for these expected patterns of gene structure, this infers you already know what those expected patterns look like for that organism. So these algorithms do, in fact, have to be trained for every genome. And this has very important implications I'll later explain. Maker's next step is to align transcript and protein homology evidence against the genome using programs like BLAST. BLAST-X and T-BLAST-X, for example, we can get sort of a homology evidence of other species from transcripts and protein, or we can get transcripts from the same species using BLAST-N. This indicates A, if region is likely transcribed, or B, if the homology indicates that this region is conserved and likely indicates that there's a gene here. Unfortunately, because of oddities with the way BLAST works, the statistics, these alignments are not as informative as they could be. They'll miss things, for example, they might miss exons. This protein alignment may be missing an exon here, for example.
Get a message.
And so you get more.
And I'll do that later.
Even more informative alignments. What we do is we polish these regions using another program called Exonerate. Exonerate does splice of site-aware alignments, so you can have much higher confidence in the intron-exon boundary. You can see here that, for example, in this protein alignment, we picked up this missing exon, and also around the splice sites, we've polished around the splice region to get a better canonical splice site, where BLAST creates these odd overhangs. So after polishing the evidence, Maker takes this entire set of aligned data and interprets it to find the most probable locations of introns and exons that may correspond to genic regions. The interpreted results are then sent as hints to these ab initio gene prediction algorithms like GenMark and Augustus and SNAP. It's sort of like Maker having a conversation with these algorithms where it sends it hints that alters their probability models so that they can produce hopefully even better models for those genes in that region. The result is hint-based gene predictions, and in the end what we have is a set of alternate gene predictions including purely mathematically based models as well as hint-informed models from these programs. Maker then identifies the one model most supported by the evidence alignment. This is done using a modified metrics from the sequence ontology called annotation edit distance that I'll explain in more detail in a moment. AED basically calculates the degree of overlap between the evidence and the gene annotations. And if you were to choose manually rather than automatically, you'd probably arrive at the same conclusion as the AED would have here. So, for example, you might notice that some of these models are missing exons or their intron isn't quite the right size for the region. And this is the one model that seems to capture, for the most part, the intron-extron structure suggested by the evidence. Next, using data from these transcript alignments, here in white from Exonerate, Maker can update gene models with respect to the 3' and 5' UTR. Because these gene predictions really only include the coding region, we can take the exon overlap here to identify probable UTR extensions to the final gene model. And finally, Maker computes other statistics that can be used for downstream quality control, such as the previous mentioned AED value that I'll explain in more detail in a moment, annotation edit distance. This describes how well the evidence overlaps the model. Then Maker also calculates this quality index, QI value, which provides basic transcript level detail statistics such as the length of the 5' UTR, in this case it's 0, fraction of splice sites confirmed by transcript alignments. Here we can see that 60% of splice sites are confirmed by the transcript evidence, and there are several other informative metrics that you can be pulled out of this. Now, finally, Maker produces all results, both gene models and reference evidence alignments in GFFF3 format, generic feature format version 3, which is a tab-based text file describing the positions of different features of the genome. You can see here, for example, we have a gene with these coordinates in the genome. This is a hierarchical format. It has parent-child relationships. You can see here that this gene is the parent to an mRNA or a transcript, and it can have multiple transcripts, and then the mRNA is the parent to multiple exons making up that transcript, as well as the CDSs within those exons. And in addition to the gene models, we also have all of the evidence alignments, such as repeat masker calls, BLAST alignments, Exonerate alignments, as well as references to the ab initio gene predictions that can all be loaded into a browser where you can perform manual annotation or curation using programs like Apollo. Maker also produces results in FASTA format for all of the models. FASTA is just a text file describing all the sequencing, either nucleotide or amino acid sequence for every gene model predicted in the genome. This data is very difficult to look at, but you can take this and push it into downstream tools, tools such as those available from GMOD, Generic Model Organism Database Project. These can include things like gbrowse and jbrowse for viewing annotations online, Web Apollo for manual curation. There's Triple, which helps create a web-based front end for distributing gene models and viewing them online, as well as other tools such as Biomart, Intermine, etc., that can be used to distribute your annotations to other groups or to mine data in a programmatic way. And in this way, those flat files can become very useful both to yourself as well as other groups by making them available over the web. Now, one of the drawbacks of using an evidence-based annotation approach such as Maker is that it takes a long time. Aligning evidence to the genome can take many hours or even days. BLAST is just not a fast approach for alignment, but it is very specific and sensitive. Whereas running a non-evidence-based gene predictor, which is purely mathematical-based gene prediction, usually takes just a few minutes. There are reasons, though, that I'll explain in a few more slides as to why evidence-based approaches like Maker are still preferable to a strictly mathematical-based gene prediction approach. But to help alleviate long run times, we've optimized Maker to run across multiple CPUs and even across multiple machines using a cluster parallelization protocol called MPI, or Message Passing Interface. This means Maker can perform analysis spread out across the network on thousands of computer processors on an academic cluster simultaneously as if it was running on a single large machine. So in the absence of a local cluster, we'd also recommend that you run this on a server, or you can even run it on the cloud. There's a protocol for running Maker in a parallelized way on the cloud by Jason Gallant that we actually link to in the Maker Wiki page. So there are many ways to get the resources you need in order to run such a computationally intensive pipeline. So how does Maker spread out its workload? To do this, Maker uses a basic annotation workflow I previously showed. Essentially, we have the input coming in. We do repeat masking and evidence alignment, followed by ab initio gene prediction, and we produce the final annotations showed here in the legacy version of Apollo. Maker, what it does is it splits the workflow into an input thread and a worker thread to do the analysis. And it goes a step further by actually dividing up individual contigs or chromosomes so that they can have their analysis happening simultaneously in multiple CPUs or across multiple machines. In addition to that, Maker can also split data up into chunks, 100 KB chunks across large contigs, and split those chunks up across CPUs and machines. You can also divide up BLAST analysis, Exonerate realignment, as well as run the gene predictors in parallel all at once while you're processing an individual contig or region of the genome. So this creates 3 separate levels of parallelization: the contig level, an internal region level, as well as a third-party tool level. So you can break your data up into very small tasks that distribute very well across hundreds or even thousands of CPUs. So this makes Maker potentially very, very fast, depending on the resources you have available. So if you were to look at the data throughput of Maker using MPI, measured on a 10 megabase region of the C. elegans genome, you can see here that the throughput of Maker, measured in megabases per hour, increases almost linearly with CPU usage. At 32 CPUs, I could annotate the entire 100 megabase C. elegans genome in just 10 hours, for example. So using a large server or a large Amazon instance. Viewed differently, using a different organism, the maize genome, which is a much larger 1.8 gigabase genome, here we're looking at the time it takes to complete the annotation given the number of CPUs. So the entire genome can be annotated in just about 2 hours if I have enough resources. At around 500 CPUs, you notice that the time required to annotate this genome starts to plateau. This is because I've hit a limit where I simply can't read the data fast enough to keep all the CPUs busy. With large analysis where I'm producing lots of output files and reading lots of data in, you can reach a point where the hard drive just essentially can't keep up. And Maker, given enough CPUs, can eventually reach that point. But you can still finish even very large genomes in just a few hours. The ability to scale up data throughput becomes very important when working with certain organisms like plants, which have extremely large genomes. Humans, for example, are known to have large genomes at 3 gigabases, but some plants, pine trees, for example, can have 20 or even 30 gigabase genomes. That can be a very daunting task if you're trying to run a evidence-based annotation pipeline.
20 gigabase genome?
Yes, 20 to 30 gigabases, very, very huge genomes. And in fact, by parallelizing one of these large genomes, we were actually able to process the 2 largest ever sequenced genomes, the 23 gigabase loblolly pine, as well as the 30 gigabase sugar pine, in just a few hours. The loblolly pine took around 14 hours of actual computation time on a supercomputer out in Texas, while the 30 gigabase sugar pine took about 30-40 hours, basically a weekend on the compute cluster here in Utah. So using tools like Maker that can scale parallelized way mean that you can still process very large data sets, even though they are computationally intensive, as long as you have the resources available to you. And there are resources available through groups such as CIBERS, as well as the National Institute of Health Exceed program, which provides access to servers for US-based researchers. And CIBERS, I know, can provide cloud-based instances to researchers both nationally and internationally. So there are ways of getting access to additional computational resources if you don't have those through your university. Earlier, when I introduced Maker, I presented it as a tool for second-generation genome projects. This is because falling sequencing costs have led to a major transition in the types of genomes being sequenced, what groups are sequencing them, and how they perform their analysis. So let me explain. Historically, because of the high costs of genome sequencing, first-generation genome projects focused on classical model systems. These are things like mouse, C. elegans, fly, for example. And these are groups that are studied by large groups of researchers. Organisms that were studied by groups that had the resources to justify expensive undertakings that was sequencing at the time. They also had the resources to pay what were essentially armies of scientists to sit behind computers and perform manual annotation and curation of the genomes. Literally dozens of scientists locked behind cubicles, just moving introns and exons around on a computer screen. First-generation genome projects also benefited from prior knowledge about the genomes. There were thousands of genome models that existed before the genomes were ever even sequenced. Pre-existing genome models allowed first-generation projects to highly optimize and tailor gene prediction algorithms for specific organisms. These pre-existing models provided a basis for later manual and automated genome annotation efforts. So what does this mean? Well, to understand what effect this had, if you were to look at how ab initio gene prediction algorithms and annotation pipelines like Maker perform on first-generation genomes, a very interesting pattern emerges. So here we're going to look at how the purely mathematically based Augustus, GenMark, and SNAP algorithms perform on the C. elegans genome. They're going to be using HMMs and prediction models that have already been optimized using the thousands of pre-existing models from the C. elegans genome. And here we see that these algorithms produce an accuracy, which is a measurement of essentially whether or not I match the models that I know that already exist in the reference, of close to 90%. That means that any given model, I will, at the nucleotide level, have a basically 80 to 90% identity to the true gene model that exists in the reference. And this is using purely computationally based methods. When I run a more computationally intensive annotation pipeline, which uses evidence alignments to inform programs like SNAP, Augustus, etc., to the location of probable exons, you see that the overall result is that we produce a accuracy very similar to what we get with the purely mathematically based models, around 88%. So not a lot of difference between the highly computationally intensive based methods and the purely mathematical based methods. And if we look at other organisms, these patterns, we see them again and again and again. The result is that there doesn't seem to be a lot of benefit to necessarily using these annotation pipelines. We see that with enough training data, ab initio gene predictors can match or even outperform annotation pipelines. So why even run a computationally intensive method like Maker? The reason is because genome projects have changed. Second generation genome projects, the kind of projects people are running right now, represent emerging model organisms. These are things like flatworms, maize, or exotic sea snails. These are novel exotic organisms with novel genome structure. They represent new experimental systems we know very little about. There's very few pre-existing gene models, usually no genetics. We therefore have no, and we also don't have, these are organisms being studied by very small groups. They don't have the large numbers or resources to be able to undertake manual curation or annotation or try and build the large gene sets that these first generation genome projects were able to use to highly optimize the ab initio gene predictors. Also, because these are small groups, the first annotation sets they produce may be the only genome annotation sets produced for these organisms. So they need to be as high quality as they can be on the first pass. So let's take a look at how ab initio gene predictors and Maker perform on these second generation genomes. Because there's no reference set for emerging model organism genomes, I can't do things like calculate accuracy as I did with the previous analysis because I really have nothing to compare to. They have almost no pre-existing models. Instead, I must either use metrics like, I can use other metrics, for example, protein domain content as a proxy for annotation quality, with the assumption being that models containing a known protein domain are more likely to be true genes, or at least more correct than models not containing a domain. So, to identify the expected enrichment of protein domains for a newly sequenced organism, I calculated the average domain content of 6 well-annotated reference proteomes using InterProScan. So as you can see, 68% of these combined reference proteomes have at least one protein domain in the genes. So as you can see, this would represent essentially the upper bounds of what would be expected to find in a newly sequenced organism. If I then run the gene finder snap on the emerging model organism Smetea mediterranea, which is a free-living flatworm, using the limited amount of training data available and perform the exact same analysis, as you can see, only 6% of the genes contain at least one PFAM domain. This is far short of what we would expect looking at the reference proteomes, and this suggests that these predictions SNAP is giving are highly inaccurate. However, when I run Maker, using the exact same data set, so Maker is not using a different mathematical model, it's just supervising SNAP. It's running the exact same HMM, but feeding SNAP hints. We go from this 6% PFAM domain content enrichment to 50% domain enrichment, much more similar to what we see with this upper bounds from the reference proteomes. This suggests that in the absence of extensive pre-existing structural knowledge, evidence-based annotation pipelines have an advantage over ab initio gene predictors. And if you remember in my first analysis, I showed you how ab initio gene predictors can perform well on first-generation genomes when optimized using pre-existing knowledge, and they can even outperform gene predictors. And in my last analysis, I showed you that those same predictors do not perform nearly as well on second-generation genomes with limited training data. I can additionally confirm those results by doing another experiment where I'm going to annotate those first-generation genomes using incorrect species parameters files. This should emulate what you would see with an either poorly trained or incorrectly trained gene prediction algorithm. So previously, when I annotated Arabidopsis thaliana, these were the results I got. So you can see that I had relatively decent accuracy in the 70s, almost 80% nucleotide level accuracy using a purely mathematically based model. But if I use instead the HMM from C. elegans to try and annotate Arabidopsis, you see that the accuracy levels drop dramatically, down to around 40–50%. Now, if I use an even more strict measure of accuracy, rather than nucleotide level, I'm going to use an exon level accuracy, where the entire exon has to be correct before I consider it as being accurate. In this case, you can see that we have an exon level accuracy of 60 to almost 70% using the mathematically based models, but it drops extremely when we use the wrong species parameters files. We can see for SNAP, for example, we go from nearly 60 to down to close to 20%. It's just 1/3 of the accuracy we had with the optimized species parameters. However, if we look at Maker's performance using the same incorrect species parameters file, so we're letting Maker use SNAP using this C. elegans file, it is able to rescue the performance. You see here, we're now getting an exon level accuracy equal to or even better than we were getting with the optimized species parameters alone. And this is using the wrong parameters. It's just the fact that Maker is able to feed evidence to SNAP so that it can alter parameters on the fly as it's running. And we see this pattern again and again and again. So in all the organisms we're looking at, when we use the wrong species parameters, Maker is essentially able to rescue the performance of the purely mathematical model by using the evidence to rescue those models, thus producing higher quality annotations that than you can get with a predictor alone, and it does this on the first try. And this is the reason why it's preferable to use an evidence-based pipeline on newly sequenced genomes, simply because we don't have the information needed to optimize the purely mathematical models with the first go. Unfortunately, de novo annotation is not the only problem facing genome annotation projects. Because of the sheer volume of genome annotations currently in annotation databases, with more being produced daily, it's not practical to review all annotations as part of regular maintenance, at least not in the short run. So quality control and data prioritization are therefore imperative to focus limited resources where they will provide the greatest benefit. Also, falling sequencing costs are providing researchers with the opportunity to revisit previously published genomes to update annotations in light of new genome assemblies or transcriptome sequence. There's also a need to integrate new data into existing databases as part of ongoing database management without actually altering genome annotations. Maker provides solutions to each of these problems. So first, let's look at quality control. Historically, gene predictions have been evaluated using metrics such as sensitivity and specificity, the average of which is commonly called accuracy, which I previously used in my other slides. Sensitivity, as seen here, it indicates how much of a supposedly true reference model did we predict. So you can see this model, even though it over-predicts, has 100% sensitivity. We predicted everything we should have. So sensitivity measures your false negative rate. Then there's specificity. This detects how much of what we predicted should have been predicted. This is essentially your false positive rate. You can see here we over-predicted, therefore we have 50% of the model is in regions we shouldn't have predicted, therefore specificity drops. And then accuracy is normally the average of the two. Here's a few more examples of sensitivity specificity. You can see here a gold standard gene. Here we have perfect accuracy, perfect sensitivity specificity, poor specificity, but good sensitivity, poor sensitivity, but good specificity because we're not overcalling. And here's a model where we have both poor specificity and sensitivity. The problem with sensitivity specificity metrics is that you have to know the correct model, seen here, before you can even begin the analysis. But for second-generation genomes, we don't know what the genes should look like going into it. So we need another way to provide similar metrics. So one idea is to use the evidence as the reference. So you can see here, if we were to collapse protein and transcript-based evidence, they will essentially mirror the intron exon structure of what should have been the actual gene, creating this gold standard evidence model that we can then use these same sensitivity specificity metrics against the final gene model to determine how closely we match the evidence. So this is called annotation at its distance. It's the same sensitivity specificity metrics where we take the average, the accuracy, and we subtract that value from 1. So in this case, AED, the 0 value means perfect match, and a 1 value means complete mismatch. So lower values are good, higher values are bad. And once again, we can see here perfect accuracy would give an AED value of 0, while poor sensitivity and specificity values give higher AED values. And if they're both have poor values, you get even higher AED values. So this is a convenient method to use evidence to try and create a numerical quality value for all your genes. If you were to then plot the cumulative distribution of what fraction of all gene models in a genome fall under a specific AED value, you have a visual way to quickly evaluate changes to evidence support for an entire genome. For example, if AED values shift to lower values for all genes, this means a better annotated genome. For higher values of AEDs, this would mean a poorly annotated genome. So better annotated genomes, the curves will shift up and to the left, poorly annotated down to the right. As proof of principle, we can use the AED to see how it mirrors other genome quality metrics, such as the 5-star rating system used by TARE, where they give different star values depending on what evidence supports a given model. We can see here in this figure that 0-star models that have no evidence support also have very poor AED curves, while the higher, as you get more stars, which means more evidence support under the TARE system, it also seems to mirror the AED system with curves moving up and to the left, meaning lower, better AED values. Here's another example of using historical data to show the utility of AED prioritization. Here we're going to use AED to classify 2 different releases of the mouse reference genome. Release 30, which is the initial mouse reference genome releases, and we're going to compare it to release 37, which is the 2007 release of the mouse genome. Here I plotted cumulative fractions of AED in the same graph, and as can be seen, between the release 30 and release 37, the annotation at a distance improved dramatically. And this shows you the effect of the efforts of the mouse community in manually curating and updating the genome assembly to greatly improve the models. This spike here in the end represents gene models that are likely false positives that have very poor AED values that were likely thrown out in between releases. The dotted curve is RefSeq annotations. They're a subset of the genes that were identified by RefSeq as being high quality, and we seem to confirm that with our AED curves being shifted up and to the left. If we want to further look at the analysis, we can bin the genes that fall in different AED regions, and we can see that genes that tend to fall with high AED values, if we look at their domain content, we see that genes with low AED content from the mouse genomes, release 30, tended to have very high domain, protein domain enrichment, while genes with high AED values, or what we think of as poorly annotated genes, have low protein domain enrichments, suggesting these are very likely false positives. We can also go a little further in this analysis, and we're going to look at what models were maintained from release 30 to release 37, and we will see that genes with low AED values were overall 86% of them were retained between releases, and high AED value models, the grand majority, 59%, were dropped between releases. So what we see here is that the AED values we produced for the initial release 30 seem to suggest that seem to show that AED seems to mimic the same decisions made by the manual curators. AAD would suggest that these are poor models, and we see overall that the curators tended to throw these models out between releases. And AAD would suggest that these are better models, and overall we see that these tended to be maintained between releases. So if I, as a small researcher, want a quality control metric that can focus my limited resources towards the genes that I will be able to curate rather than trying to curate everything, AED is a nice metric because it helps identify the subset of genes that are in most need of manual review. Another of the issues beyond de novo annotation facing genome projects I mentioned earlier was updated and revising published annotations in light of new evidence and assemblies. Maker can do this by taking legacy annotations that may be from a different group, or they may be curated sets that are maintained by individual laboratory. There may be also multiple versions of the genome by competing groups, and what you would like to do is you would like to combine those data sets together with evidence and updated ab initio gene predictions to create a merged, curated, current assembly that has a single non-redundant annotation set that merges all of the data. So Maker has a way to do this for you. We've actually done this previously with the maize genome. Here is a proof of principle analysis we did with just chromosome 4 of the maize genome, where we look here, we have a gold standard reference set of manually curated genes from the existing reference. We have what Maker would have produced if Maker was allowed to predict models all on its own. And then we have the current reference data sets represented by this line. And when we allow Maker to run by giving it the current reference set together with the new models, Maker can merge those data sets into a single new data set, which has a better overall AED curve, where it both maintains some of the previous model as well as updates models wherever it finds that the evidence best matches the new models. And you can see here, for example, many of these models at the end, which have low AED values, probably very limited, or if any evidence support, were maintained in this evidence combination run, as shown in this AED curve. So this is one method you can use to update your genome annotations using Maker. Another issue facing genome projects is the ability to integrate new data into existing databases as part of ongoing database management. So you don't want to alter the gene models, you just want to add evidence and maybe annotation statistics to those gene models. You can also do that using programs like Maker. Here, for example, we have 8 ant genomes, and we have produced AED curves by aligning evidence to those reference data sets and just pushing them through Maker without allowing it to modify the gene models. This is nice because now we are able to identify the genes that would be best served by manual curation. We also perform the domain analyses I previously showed, where low AED value genes tend to have higher domain enrichment than higher AED value genes, suggesting that these are either genes that are false positives or may even be organism-specific, but this is where I would focus my manual curation efforts and review. If I look at orthology between the genomes in different AED bins, you'll also see that genes with low AEDs tend to have high orthology, with the average gene having 4.4 orthologs in other species, while genes with these poor AED values tend to have very few orthologs, suggesting they're either false positives or they are species-specific genes that just don't have a lot of evidence to support them. Once again, genes that you might like to look at manually. Okay, so now I've given you an overall view of the annotation process, and I've explained what Maker can do, and just go and tell you basically what's available as far as how Maker's grown during the years. So the initial version of Maker came out in 2008, and since then we've updated it to Maker 2 and Maker P to give it parallelization, as well as support for RNA annotation. And now we've moved on to Maker 3, which also has the ability of users to add weights to their evidence data sets. See, if you don't have much confidence in a protein data set you're using, you can give it a lower probability weight in the final models that Maker's producing. And all of these versions of Maker are command line tools. And unfortunately, command line tools are very hard to show in presentations. So we've also developed a web-based interface version of Maker. It's sort of a toy version of Maker where I can show you what Maker does online. So to close out my presentation, I'm just going to give a quick overview of what an annotation project process looks like using a web-based interface. So you can go to www.andellab.org and access Maker web annotation service to push through small data sets to see what how Maker works on a small region of your genome. So to do this, we click on a new guest account, and we are going to go ahead and log in, submit a new job. Here we're going to slide down and select the assembly we're going to use. For the command line version of Maker, you simply provide a file in a text-based configuration file. But here we're going to just select the file, and you can see this is a standard FASTA-based assembly. Then we scroll down, we're going to select transcript data set we're going to use as evidence. Then we're going to select also a protein data set. This can be something, it doesn't have to be from the organism you're using. It can be from a neighboring species, closely related species, or from a curated data set like sweet uniprot, Swiss prot that contains a large number of species. Then we're also going to pick the gene prediction algorithm we've trained on the training data available. And we're going to run that through Maker. Maker then produces results in GFF3 format that are hard to look at, but if you push them into downstream programs like gbrowse, you will get a visual representation of the gene, which is nice here. You can see the introns and exon structures displayed nicely in a browser alongside with the evidence alignments we see from Exonerate, or you can view things in jbrowse, which is just an alternate viewer to gbrowse. Once again, we see the transcripts as well as the evidence from proteins and transcript evidence that suggests and supports each gene annotation. And then we can move on to something like Apollo. This is the older desktop version of Apollo. Same view, but the difference with Apollo is you can manually move around introns, exons to make changes to the final GFF3 file. Or we could push this into other types of downstream analysis. For example, this is a tool from the sequence ontology which produces downstream metrics and figures. For example, here we're looking at a histogram of the CDS distribution for genes through the annotation set. Or you can download a data set and get the raw data, and you can then use in other tools to perform any number of other kinds of downstream analyses. Once you've have gene models, you may also want to do post-processing of the annotations, and Maker contains a number of accessory scripts that make post-processing possible. Here we've wrapped those accessory scripts so you don't get to see what they do, but you can run BLAST, for example, against uniprot, Swiss prot to identify putative orthologs or homology to other genes and add that information to your GFF3. You could also use interproscan to analyze the FASTA files to identify protein domain content of genes, as well as the Go terms that might be associated with a given gene. You can also rename those genes using an organism type prefix, and Maker comes with tools that will help you integrate the data from BLAST, interproscan, or an updated naming file for your gene models into your final GFF3, and here we'll just let the web interface do it for you so you can see what the overall result is in your downstream annotations. Here we're going to look at it in gbrowse, and you've seen the gene models have now changed. We have much nicer prefix names. We have putative orthology to each gene, for example, similar to that one synaptic vesicle membrane protein. You also have the interpro domain locations now identified in the genes. And if we click on an individual gene, we can have all the information on all the interpro domains, PFAM domains, Panther domains that a gene contains, as well as the Go terms associated with those domains. So we've been able to enrich the overall content of the gene annotations with a few simple accessory jobs run post-Maker. If you want information on how to run those accessory scripts or Maker itself, you can go to the Maker Wiki page. Here's an example. Going to the Wiki page, you can see that there are a number of tutorials and classes that contain test data sets as well as step-by-step instructions on how to annotate a genome post-processing. And if we click on the first one, you'll see that if we scroll down here, we can find post-processing annotations. I'll click on that, and this would be an example of the command lines that would have produced the exact same result as I did via the web-based interface. So go to the wiki, and you can get an example of how to run Maker, and like I said, step-by-step instructions to do all the steps that I quickly showed you with that toy example. So that really brings me to an end of what I wanted to show you today. The last part I rushed through a little bit, but feel free to ask any questions you want, and I'll try and expound on the details on what was going on with and how Maker works. I'd like to acknowledge Mark Yandel, my professor and boss. He originally had the idea of Maker from his previous work at Solera Genomics. Barry Moore, our lab manager, was heavily involved in algorithm development and maintenance, as well as lab alumni Michael Campbell and Daniel Entz, who were involved in Maker development testing. Some of the data and figures I showed you came from Michael Campbell, for example, and both are still involved in the Maker user list and answered user questions very often there. So now, I'd just like to thank you for your patience in listening to this presentation, and I'll take any questions you have.
Wonderful, Carson. Thank you so much. We've received several questions. I'll ask before this one, are you aware of the current EBP project?
EBT, I'm not.
Okay. EBP is, the goal is to sequence all living things in the world. So given that...
Eukaryotes.
Okay, I'm sorry, all eukaryotes. The bacteria are on their own. So I suspect they'll sneak in, and they have other projects going for them. So anyway, so for all eukaryotes, the goal is to sequence all of them. So given that, do you believe that the current computational analytical tools and power required for Maker are going to be accurate to meet that need?
Given enough time, you could do it. Of course, like I explained, these can take very long amounts of time. We are trying to figure out ways where we can sacrifice specificity and sensitivity to still get similar accuracies, but maybe slightly less so, by using more modern alignment tools. Everything I showed you, for example, was BLAST. BLAST is 80% of our runtime. But there are short-read hash-based alignment variations that can produce alignments for longer gene-sized reads and do it very rapidly, 4 to 10 times faster than BLAST, for example. And we're currently working on integrating those approaches into Maker, and that would bring run times down by maybe a factor of 10. And using those tools, like I said, you do sacrifice sensitivity somewhat, but if you have a large number of genomes, that's the price. If you can either take a long time and use a lot of computational resources, or you can process 10 times as much data and take a slight sensitivity hit. And I think that's probably what's eventually going to happen, is you just need to accept that annotations aren't perfect, but we have a lot of them. So you as a researcher will still need to go in and at least do a manual review of what evidence is supporting a given annotation. And in that way, you'll have a better understanding of whether or not you want to trust it for the experiment you're using.
Great. And so with that would also come the ability to simply run with default parameters, is my guess, in order to be able to automate these things. Do you feel like the default parameters, again, would be able to handle that?
I think so. Your default parameters without much optimization will get you a 70 to 80% nucleotide accuracy, which is pretty good. I mean, it takes a lot of effort to add a few percentage points onto that. You don't get up to those 90% accuracy levels without a lot of optimization and manual curation. 80% is really about the highest you're going to get with computational methods, and it's almost like cell phones. For a while, they had good cell phone reception when they had these big refrigerator-sized cell phones, and as they started shrinking, cell phone reception got worse and worse, and they gradually built up the infrastructure to where cell phones are getting good again. So genome annotation is sort of that way, in that the first set of annotations were very nice and pristine. The coming genomes are very poor in quality, but as we get better evidence data sets like mRNA-seq, they're getting better. So I think we'll eventually get to a point where we'll have a nice default 80–85% accuracy genome annotations just from a first pass-through with good transcript evidence.
Great. We also had another question. Is the Cantrell Genome Research 2008 paper, specifically Table 2 with the Maker Quality Index Summary, the best reference for the Maker QI string? This user noticed that sometimes the number of exons in the QI string is different than the number of exons in the GFF file.
I'd have to look at that. It should be the proper reference. Depending on which value you're looking at, I know some of those values use every exon, and some of those values are only using the coding exons, I believe.
Great.
But they can point to me a specific example and which part of the QI they're looking at. Preferably send it to the maker list, and I can take a look at the model and tell them whether it's a bug or whether it's just that I need to give a better explanation on that specific QI parameter.
Great. So many of us tend to assume that more is better when it comes to evidence for predictions, but this can lead to, like you said, long compute times. How would you filter large volumes of evidence to produce alignments with reasonable compute times? For instance, is it better to have just tons of RNA-seq, or is there a way to filter that down to get the most out of it with a shorter compute time?
I recommend what you can do is you can annotate just a very small region of your genome, pick one of the larger contigs, and only run that contig. That will give you a very short computation time, and you can put that up in a browser. What you'll often see is that when you look at the data sets you've chosen, you'll see some of them might have, if it's a protein data set from another organism, you might find that it's very poorly annotated, and you just want to throw that data set out completely. Your mRNA-seq data, you don't necessarily need 20 mRNA-seq data sets. You could combine them all into the single run and perform an assembly of the collapsed mRNA-seq, which should provide the assemblies to give you a nice collapsed assembly with lots of coverage to provide you the major transcripts expressed in that genome. If you are interested in the alternate spliced versions, then yes, you will have to produce assemblies for different tissues and different treatments to try and capture alternate splice forms. But if this is your reference annotation, for the most part, you really only want the one model representative for every gene, at least for the first go. And I would also suggest using data sets like UniProt, SwissProt, which tend to be good for the overall coverage of the genome, so you can get everything from protein-wise in a single data set rather than having 10 related species. You could use something like single related species plus UniProt, SwissProt. Also, if you have a faster way to align these to your genome, you can provide GFF3 alignments that you've already produced, so that Maker doesn't have to run these algorithms, such as BLAST. It'll just believe the alignment you give it. So Maker has a GFF3 alignment pass-through option that I know some groups use because they produce their own alignment using their own algorithms.
Okay. I think you've answered a lot of these that have come in. Given Maker's ability to pass evidence hints to evidential predictors, would you still suggest taking the time to train predictors like SNAP? And if so, what criteria would you use to know that the training has been sufficient, the bootstrapping of training has been sufficient, and you're ready to just go ahead with a run?
What I've seen is I would always train them at least one round, perhaps two. One of the easiest way to get the first round of training is using programs like, there's the older Sajima program, but now they have BOOSTCO. BOOSTCO will identify very highly conserved genes in your genome and provide training set for you that you can use with SNAP, or it can pre-train Augustus for you. And then by doing just a first pass with Maker, you can do a first round of prediction and Bootstrap. The best way to know whether or not that Bootstrap training was effective, if your ab initio predictor now by itself is producing models similar to what it produces inside of Maker, then your Bootstrap training for that algorithm was effective. A nice comparison would be using a program like Eval from the WashU. It's a program that can pair annotation data sets. So if you, for example, were to run Maker with Augustus, where you don't provide any evidence, and you run it again where you do provide the evidence data sets, and then you compare the two annotation data sets using Eval, it will give you the accuracy levels, the sensitivity, specificity between those two data sets. And if you find that they're relatively similar, then 70–80%, then you can say, okay, I've well-trained my algorithm because I'm getting a 70–80% eval rating. And if you're getting something much lower, then you probably either have a poorly trained algorithm or the algorithm just will never work in your species. SNAP, for example, won't work on some mammalian species. It just never performs well. And it's best to just drop it from the analysis. GenMark performs well on fungi and many gene-dense organisms, but many mammalian species doesn't perform well as well. So either you can look at these things in a browser to get that sense, or you can do, like I said, an automated view where you just do the first Maker run with your training set without evidence, second run with evidence, and if you have a similar result, then you know your model's trained well, and the evidence should just be giving you a minor boost above what Augustus or SNAP is producing on its own. Great.
And what's the best way to include evidence from full-length cDNA sequencing, such as PacBio or Oxford Nanopore?
So with Maker, the best way to do that is currently through assembly. If you can do read-based assembly to create what would essentially look to Maker like a cDNA, then Maker is able to align it, because the alignments, Maker wants the alignments to have an intron somewhere in the alignment, and that actually improves the accuracy of placement of the reads inside the Maker algorithm. If Maker isn't hip to all of the new potential methods that are available out there with short read and long read sequencing, so sometimes it may be best if there's something specific about your data that Maker isn't capturing, you can produce the alignments yourselves using other programs, StringTie, for example, where instead of producing the assembly, you produce basically an alignment in GFF3 format that Maker can then consume. And it can use that alignment to identify the locations of introns and exons. The one thing to avoid with the mRNA-seq data, especially the short read assemblies, is that they can falsely merge across genes. In fungi, for example, UTR do in fact overlap between genes and opposite strands, and your mRNA-seq assemblers will assemble two genes into a single transcript. So using tools like Trinity, which are aware of that, they have a Jacquard indexing option that will try and break those false merges apart. Things like that can help. There's also a post-processing tool out there called Diffusion, and if you use that tool to post-process the Maker gene annotations, it will break apart any genes that it thinks were merged because of false mRNA-seq merging.
Great. Maker, Carson, this has been a really wonderful webinar, and given the time, I think we're going to go ahead and end here. We're going to have a webinar next month from the NCBI group that I hope you'll all attend as well. I just want to thank you again, Carson, for such a wonderful webinar, and I hope everybody has a great day. Thank you.
Thank you very much, and if people want to send me questions through the Maker email list, they can do that as well, and I'll try and answer them.
Fabulous. Thank you again.
Thank you very much.
Thanks.